Good morning, dear students. Today we are going to discuss about this action potential, right? So this action potential is a very important topic as far as exam is concerned. University exam will be having either LAQ or a short note on this action potential. It is most commonly asked question in the general physiology section, right? So this at the end of this session, the learner must be able to how to define the action potential, then what are the properties of action potential, then ionic basis of each phase of action potential, and its application in the clinical physiology. So this action potential, so this action potential, what is this action potential? Basically it is, so as the name suggests, action potential, that means there is some action which is happening in the excitable tissue. So excitable tissue is defined as a tissue which is showing response when it is stimulated. So that is the definition of excitable tissue. So there are mainly many types of excitable tissue in the human body. So first and most important excitable tissue is the neuron, and next is the skeletal muscle, and finally the glands. So these tissues, they will respond in the form of, that is a nerve, by showing this some releasing of these neurotransmitters, which may be excitatory neurotransmitter or the inhibitory neurotransmitter, right? And then muscles, it shows response in the form of contraction, mechanical contraction in the muscles, and the glands, they will release their secretion into the circulation when they are stimulated, right? So these are the three important types of excitable tissue in your body, and this action potential, it is responsible for the generation of this excitation. That means they will excite, they will show response only by means of generating the action potential, right? So this action potential, it is a key, it is a basic phenomenon which is nothing but the communication between the neurons. So neurons communicate with each other by generating the action potential. So many functions of neurons are there. For example, it is the perception of color. Your photoreceptors, they are responsible for recognition of color. So there is generation of action potential in the photoreceptors. That means you will able to recognize the color only when there is action potential is generated. Then sense of hearing, you perceive the sound, you can recognize the person on the basis of having memory of recollection of memory regarding that particular voice. So you can recognize the person by hearing the voice. Then perception, whether it is touch, pain, temperature, you can perceive that thing. Sense of emotions, so whatever things are happening which are related to the neurons, either by releasing the neurotransmitter, it may be excitatory or inhibitory, which is happening only because of generation of action potential. So that's why this action potential is very important to learn conceptually, right? So this action potential, so by definition, so action potential is nothing but it is reversible, transmissible, and conducting change in the resting membrane potential. So student must be able to understand what is resting membrane potential, because the generation of action potential lies in the generation of resting membrane potential. So when if only their resting membrane potential is there, then only action potential can be generated in that particular tissue. So what do you mean by resting membrane potential? This resting membrane potential, as the name suggests, resting membrane potential, that is, it is nothing but the potential difference which is existed across the cell membrane under the quiescent stage, under the resting state. It is called as resting membrane potential. So different excitable tissues, they will be having different resting membrane potential. For example, nerve, it is in the range of -70 to -90 millivolt, right? So like that, each and every cell will be having definite charge which is developed across the cell membrane, and that charge, that potential difference, that is called as resting membrane potential, right? This resting membrane potential, it is developed because of unequal distribution of charged ion across the cell membrane. For example, the sodium ion which are present in excess quantity on the outside, that is the extracellular fluid, as compared to the intracellular fluid, right? So that's why there is a more crowding of positively charged ion outside the cell as compared to the inside of the cell. So that's why the cell is being negative inside and positive outside. So that is the basic theme of generation of resting membrane potential. It is not just only the sodium ion which is responsible for generation. So there is an interaction which will be covered in the separate lecture which is related to the resting membrane potential. Today we will discuss only to the action potential. So action potential is nothing but the change in the resting membrane potential, which is transient, which is conducting, and which is reversible. Also it is self-generating change, right? Now we'll further continue our discussion. So properties of action potential. So properties, as it is transient, that is short lasting, transmissible, self-generating, reversible, it follows the all or none response. That means whenever the action potential is generated, the amplitude of action potential is to its maximum extent, or it is not showing response at all. That is the basic understanding of all or none response. Then it is having some refractory period. Then infatigability, you can generate one, two, three, many action potential one after the other. That is infatigable phenomenon, right? Now, what is the basis why action potential is generated? So, basis lies in the presence of definite ion channel across the cell membrane. Now, there are various electrolytes, various charged molecules are present both inside as well as outside the cell membrane, right? So, there is a movement of this charged ion. So, sodium, potassium, then calcium, chloride, magnesium, all these are charged ion, and each ion is having dedicated ion channels. So these ion can move across the dedicated ion channel only, and when the gates are open. So some channels are, these are called as leaky channel. That means they are more leaky in nature. So they will allow the movement of potassium and the sodium in the leaky manner. Then again, some channels are the voltage gated channels, right? So voltage gated channels means what? So when the gates of these channels are open, when there is change in the voltage. Some channels are ligand gated channels, that is attachment of some chemical to the receptors, and then only this channel will open. Some are ATP driven pumps. So because of interaction of these channels, there is a movement of ion across the channels. So that's why there is reversal of polarity or change in the resting membrane potential, which is for short lasting time and which is again responsible for generation of action potential, right? So there are certain terms which are used in this discussion, that is a polarized cell. So what do you mean by polarized cell? So polarized cell is the cell which is in the resting state. As I have told earlier that there is a unequitable distribution of ion across the cell. So positively charged ions are more on the outside as compared to the inside. So that is more positivity outside, less positivity inside of the cell. So generation of potential difference across the cell. So that is, the cell is said to be polarized in the resting state. Then what is depolarized cell? So that is a reversal of polarity or change in the polarity, right? So it is called as depolarized cell. Repolarization, again attainment to the polarized state, that is called as repolarization. Then what is hyperpolarization? So cell become more negative as compared to the previous initial quiescent stage. So that's why it is called as hyperpolarization, right? So phases of action potential. So these are the 8 stages. So when we study this nerve fiber, so Hodgkin and Huxley, these are the two novel scientists. They are the Nobel laureates. So these scientists studied the giant quid axon for the purpose of this generation of action potential. What are the phases of action potential, right? So in this PowerPoint, you will see on the left pane of this slide, you will see this cylindrical structure. These are the neurons. On the upper pane, that is the part A, you will be having some recording instrument which is responsible for the recording of action potential across the cell membrane or any potential across the cell membrane, right? So this is the cathode ray oscilloscope. So when we put electrode inside of the cell, so these 8 stages are present when we studied outside of the body. But when there is generation of action potential, it is broadly divided into the only 3 stages. First, that is the depolarization state, then followed by the repolarization, and then hyperpolarization. But when we studied under the study material, so you will be having this resting membrane potential, that is the initial baseline phase, then stimulus artifact, then latent period, firing level, overshoot, spike potential after hyperpolarization, right? So these are the stages of action potential. So this is the graphical representation when we studied some animal, right? So we will get this kind of graph. So in this graph, Along the x-axis time is plotted, along the y-axis potential difference is plotted, and there are various certain stages of this action potential. So A, B, C, D, E, and finally the F, right? So this red color graph, it will indicate the change in the resting membrane potential. So you can see, so the change is very transient. So the time is plotted in the milliseconds. So trains that change is very of transient time. So within fraction of millisecond, the change is occurring. So A is the stimulus artifact, right? The B is the firing level. So from beyond the firing level, there is a rapid upstroke and the rapid downstroke, and followed by that again there is a slow phase, and then it is the graph is again moving down of this baseline. So that is the hyperpolarization phase. We'll see, right? So ionic basis of action potential, what are the reasons behind the each phase? So upstroke of this graph, that is from A towards the C, so that will be the depolarization phase, and from C towards the E and F, that will be the repolarization phase, right? So that depolarization phase will be having this firing level. So the basic reason behind this generation or the formation of this depolarizing phase is that, so it is the opening of sodium channels. So sodium channels are present on the cell membrane. This channel will open. So initially these channels, few channels will open, and they will cause the change in resting membrane potential towards the positive direction. So initially few channels are open, but when more and more sodium channels are getting open, so change will be more rapid beyond this firing level. So firing level is of having amplitude minus 55 millivolt. So at this potential difference, many of the sodium channels are open at once. So that's why there is rapid upstroke, which is certainly beyond the zero potential, and this phase is called as spike potential. It is also called as overshoot. So after the overshoot, we'll be having the rapid repolarization. So depolarization reason is opening of sodium channel, and for the repolarization there will be the opening of potassium channel, right? So these potassium channels, they will open, they will start opening right from here, but the peak of opening of potassium channels is at this potential difference level. So at the level of plus 35 millivolt, this potassium channels fully open, all the potassium channels are get opened. Because of opening of potassium, there is rapid efflux of the potassium ion. So in the depolarization, there is rapid influx of the sodium ion, while in case of repolarization, there is rapid efflux of the potassium ion. So again, the potential difference is reaching towards the negative value. So initial repolarization is rapid, followed by that is slow repolarization, because potassium channels are slow to open, and then again they are slow to close also. So that's why it is reaching beyond the resting membrane potential value. It is called as after hyperpolarization, right? So you can see here diagrammatic representation. So sodium channels is shown in this figure. So this is the membrane section. So sodium channel will be having two gates, activation gate and inactivation gate. So under resting state, activation gate is always closed. So when the small change is happening, so when the small change is happening towards the positive direction, so these channels, they will tend to open more and more. And when these channels are open, so activation gate is open, so sodium will be moving from outside of the cell to the inside of the cell. It is responsible for development of depolarizing potential. So same thing happen for the potassium. So but for the potassium, it will be having only one gate which is present on the inside of the cell. So when this will be activated, so potassium will be coming out of the cell from inside to the outside, which leads to development of repolarizing potential, right? So if you compare this membrane permeability of sodium with the potassium, so initially, so when sodium channels are open, so they are very, during the depolarizing phase, so conductance of sodium is more as compared to the potassium. As potassium channels are leaky channel, they are slow to open, so they will take time to attain the maximum level of conductance across the potassium. But sodium conductance is increases in the exacerbating manner, so it increases rapidly and it decreases also rapidly. But potassium conduction, it increases slowly, maintains a plateau, and then again it comes down again the slowly. So that is the comparison of conductance of sodium and potassium across their respective channels. So thank you very much.